Welcome back. This is part 2 of the chapter 18 flip, and we are picking up looking at eukaryotic control in transcription. So we're looking at enhancers. Enhancers are going to be areas like you see on the screen that increase transcription, and you can see that they aren't necessarily right next to it's not the same structure as an operon. Excuse me. So notice we've got our enhancer way upstream here that's going to basically bind to this mediator protein complex and facilitate polymerase binding and just make the transcription happen easier, make the transcription happen faster. You see we've got our promoter still with our tau box that the polymerase is looking to bind, but it's just sort of a lightning rod for to help bring the polymerase and all the transcription factors together. So it's going to allow folding. You see it's a complex. It's a coordinated kind of thing. So see, we've got these different we've got these different control elements in different cell types that are going to cause different genes to be expressed based on which enhancer and which folding pattern we see. If we get these available activators, we get these available activators, we see the folding pattern is different, so a different protein is expressed in liver cells versus lens cells. After transcription, we can do some things like alternative processing, the alternative splicing. That's something we've looked at a little bit before. So could be introns and exons. Again, basically, it gives us a different combination of possibilities. You see here we've got 5 exons, and you guys have seen this picture before. Depending on how we splice the RNA, we're going to get a different end result. The one on the left there, we've got 1, 2, 3, 5, and the one on the right, we've got 1, 2, 4, 5. So form controls function. Those two proteins are going to do something different. Another way to look at it, you see maybe it's not, maybe this segment is something special. We get this strand, we cut it out, we get same kind of thing. But in this example, BCLX, that's 2 and 3 together, is going to inhibit apoptosis. Oh, sorry, I have that backwards. This is going to inhibit apoptosis. This is going to trigger it. So they have different and opposite effects based on what's in the middle. Is it going to cause the cell to die or not? Big topic in biology, like it says on the screen. About 60% of our genes are estimated, and that's an ever-changing number. But this goes back to the definition of a gene down here at the bottom. One gene does not equal one polypeptide or even one RNA strand. That's why our definition is a segment of DNA that makes something that does something. A couple of other post-transcriptional control points. RNA transport, we have to physically move the RNA into the cytoplasm. So that process could be a checkpoint. And then eventually the RNA is going to start getting broken down. And that will stop transcription. So when we look at translational control, that's largely going to be limited by the availability of the stuff. Is it going to be the initiation factors that start the process, those helper proteins kinds of things that we talked about, or is it going to be the tRNAs themselves, which tRNAs are available, and those kinds of things. So you really want to look at Chapter 17, and anything that needs to be present for translation is a potential limiting factor, something that could limit the process. So after we make the protein, we could control the protein itself. So this would sort of be post-translational controls, and that's where maybe we're done with the protein and we don't need whatever it's doing. Maybe that enzyme is no longer needed. So what do we do with it? Well, we can change it. We can turn it on. We can move it somewhere else. We can engulf it in and encapsulate it, or we can break it down. So you see degradation here. There are these things called proteasomes that are going to use this ubiquitin to mark a protein. Ubiquitin floats around, and if it connects to a protein, it's sort of like if you've ever parked downtown anywhere and they have a parking attendant that walks around and marks your tire so that a police officer sees it and they see you got 2 marks, they know you've been there for 2 hours. Same kind of concept here. You've got 3 ubiquitins, so you must have been around for a while, so let's break you down and get you out of here. That's kind of the idea there. We also have those RNAs that I mentioned when I was a student. We learned that there was junk DNA, and now we know far better than what we knew back then. We have this non-coding RNA that's not translated into protein, but appears to be involved with regulation. Different RNAs are going to be involved in different places. You see we've got these microRNAs or small interfering RNAs that, again, made from RNA molecules. It's diced into double-stranded segments. So they're small strands, or they're small sequences. They're some of those some of the introns and things like that that are used in regulation that we're only now being able to figure out exactly how they work. RNA interference basically can interact with the mRNA, could block translation. Sometimes they physically block translation. Sometimes they're going to impede the mRNA. And like it says there, a high percentage of our DNA produces that regulatory RNA that once upon a time we called junk DNA. So morphogenesis is a really good example of gene expression control. So think about how an organism is going to go from a single-celled zygote into a multi-cellular organism. That's going to be a lot of different expression at different times. So some of these things are already in the egg. These cytoplasmic determinants are those chemicals that signal embryo development. Notice there that mom makes those, the embryo doesn't. So the egg gets a head start thanks to mom's work. So you see the unfertilized egg cell, 2 different cytoplasmic determinants. The zygote is formed. The first cell division happens through mitosis. In the zygote becomes an embryo, and we get a lot of the triangles on one side. That's going to become one cell type. The ball side or the sphere side is going to become a different cell type. Then we get this process of induction where we get cell-to-cell signaling. Think back to induction when we talked about it in the fall. That's going to give position and clues basically that, hey, you're supposed to be a nerve, you're supposed to be an arm. I'm being this, so you be this. And that's sort of what you're seeing. We're getting a message, a ligand that's going to induce a signal that's going to cause a transduction pathway that goes to the receiving cell's nucleus and manipulates expression. We've learned a lot of this from Drosophila studies. And one of the big things that came out of the Drosophila work was these Hox genes. And they're these master regulatory genes that have, you see, they've got these homeobox sequences that control the body plan of an organism as it develops. It sort of controls what becomes an arm, what becomes a thorax, what's an abdomen, what's a head. Also gives these body axes. What's the top? What's the bottom? What's the back? What's the dorsal? What's the ventral? I skipped over this quickly. Evolution is tied to gene regulation. If you mess with these homeotic genes, you get a different result. You can have two sets of wings, or you can get an abdomen where the head goes. You can manipulate those homeobox genes, and you can mess with the body plan of an organism. Next example is showing what happens as it goes from egg to larva. We end up with these nerve cell, egg cell, those types of things. We end up with these homeobox genes creating different segments, even in other species. So it's not just unique to the Drosophila organism itself, it's true of all organisms. So I know I stumbled through that a little bit. I apologize if that's at all confusing. But here's a picture of where there's been a mutant or it's been modified in some way. Notice you get legs where antenna should be. So something is messed up with the body plan. that homeobox gene has been messed with. Examples, Bicoid organism, Bicoid is a gene that produces this protein that basically says you're going to be a head. And organisms like organisms that pupate, like how a moth turns into a butterfly, they use Bicoid as well. And you see that there's a concentration of Bicoid here, and that leads to a gradient where this large amount of Bicoid causes the head to be produced. And that's going to be true in butterflies and things like that. That Bicoid is going to be dense, and it's going to, as the pupa hangs, it's going to sink down to the bottom, and you're going to see a concentration gradient. So the head of the larva develops where there's a lot of Bicoid, and where there's not much Bicoid, that's going to be a tail. It's a chemical determinant that's going to mess with transcriptional control and translational control. If there is no Bicoid gradient, you can get two tails. So if you artificially inject Bicoid into both, into what should be the tail region, that can cause the larva to be born, for lack of a better term, with two tails. And that's what you saw here, tail and tail, where it's been mutated. So there certainly are other genes that control the development of different axes and different segments of the body. I just focused on those two because those are the two that tend to be tested on most frequently. Of course, anytime we're talking about replication and control, we need to think about cancer. We've defined cancer as the loss of control of cell division many times. But there normally is a balance between the gas and the brakes, the growth stimulating pathway and the inhibiting pathway. But in cancer, we lose that balance. We're all gas, no brakes. So there are genes, some cancers are genetic, and some you don't inherit the cancer, you inherit a predisposition. So you're closer to having the wrong form of the genes. Those are called proto-oncogenes. If they get the wrong mutation, it becomes cancerous. And that's going to happen in some specific, we have some specific sequences. You've probably heard of BRCA. That's going to come up shortly. But if the gene happens to get the wrong methylation pattern or acetylation pattern, or there's a skin cancer mutation, something along those lines, that's going to lead to an oncogene that causes cancer. We have these tumor suppressor genes. P53 is one that I would make sure that I'm familiar with. That one comes up frequently on the AP Bio exam. But they're going to be genes that turn cell division off. RAS is a G protein that we talked about in cell signaling. When it mutates, it causes an increase in cell division by overstimulating those protein kinases. There are several of those mutations that are known that each mutation leads to a different form of cancer. So you see here we've got our G protein. That should look familiar to us. If RAS is mutated in the wrong way, that's going to lead to protein kinase. Basically, it's saying, hey, go, start dividing. And that's obviously a contributor to cancer. So again, the specific type of mutation and the specific location of the mutation is going to dictate what kind of cancer you have. Again, P53 is a spell checker. When it's damaged through cigarette smoke, that's a really common mutator of P53. It doesn't work. Again, it's our spell checker, and we disable it. That's going to cause cancer. It's going to basically let mistakes slip through the cracks, and it won't recognize damaged cells and cause them to die off. It'll just contribute to cancer. That's sort of what you're seeing there. This one triggered by UV light causing the DNA damage. I kind of mentioned skin cancer earlier. Causes the P53 to change shape, and again, form controls function. We know about carcinogens, probably don't need to talk about that. But multi-step hypothesis tells us that usually one mutation isn't cancer causing. Usually we need 4 to 5 mutations, depending on the type of cancer. We need a bunch of mutations. We need a bunch of things to go wrong before we see cancer. Our body